All right, so I just want to review all of the stuff that we did before break. Don't mind the playoff beard. It's your sneak peek. I got it going. Hopefully, we get to wear this until February. Anyway, depends on the year. If this is the year that I recorded this, then hopefully the Bills keep making it. I don't know. Whatever. So remember, um, when we're talking about how do we actually create proteins, we always start with the DNA. The DNA is the blueprint, that's the recipe book. We then transcribe that DNA, we turn it into RNA, right, through the process of transcription, and then we translate that. And that's what our main focus of today is going to be, right? I just wanted to review everything because it's been a while, right? So let's talk about, let's see, the process of transcription first. Remember, a gene has three parts to it. It has the promoter, it has the coding region, and then it has a terminator. The promoter is the spot. Remember, you find that with a Tata box or a Pribnell box in bacteria. And basically, that's where RNA polymerase, which is the copying enzyme, attaches. Remember, bacterial genes do not have introns, where eukaryotic genes do, and those introns get spliced out, and the exons get kind of put together into a proper coding mRNA molecule that's a mature mRNA. Let's see here what I want to talk about. Remember, so when RNA polymerase binds, it always synthesizes the new strand in the 5' to 3' direction. So it slides along the old strand in the 3 to 5, synthesizing the new one in the 5 to 3. Once it hits a long chain of A's, it's going to fall off, and we have now copied the RNA molecule. So this is showing that process. Remember the way it works. It always reads the template strand. The template strand is the one that runs 3 to 5. And then it always reads it as the opposite. So T pairs with A pairs with U, C pairs with G pairs with C, from the template strand to the RNA. Remember, the coding strand will be the same as the RNA molecule, except for all T's will be replaced with U's. Let's see, what else do I want to do? Oh, remember, so in eukaryotes, there's three modifications you have to remember. There's a 5' cap, a poly A tail, and then the splicing out of the introns. So mature mRNAs always end up being a little bit smaller than the actual RNA that was initially transcribed, or what's called the primary transcript. Because even though you do add a 5' cap and a poly A tail, usually you remove a lot more sequences through the splicing of introns than you actually gain by adding a 5' cap and a poly A tail. And remember, the purpose of the poly A tail and the 5' cap is to increase stability, and it reduces the it reduces the chance of degradation. And basically, the longer the poly A tail, the longer the RNA will stick around, which can increase how much protein is made from that mRNA. So the spliceosome is the thing that splices out those introns and cuts them out, and then puts together those exons into a mature mRNA, which has the 5' cap and poly A tail as well. And the spliceosomes are made up of both RNA and protein. Remember, I can tell this is prokaryotic because both transcription and translation are occurring together. Eukaryotes, that is not possible because transcription happens in the nucleus and translation happens outside the nucleus. So if they're occurring simultaneously, we always know it has to be a prokaryote. And I will ask a question on the test that asks that specific thing. Also remember, most times exons code for specific domains within a protein, and they usually have a specific purpose. So like if this is a receptor, domain 3 codes for the ligand binding site, domain 2 for the part that spans the membrane, and domain 1. So there's probably a bunch of hydrophobic amino acids, right, that can go through the membrane core. And domain 1 would be the part that speaks to the next step in the transduction pathway, whether that be a G protein or something else. And if you mutate these different domains, right, within the genes, you'll code for different parts of that domain, and that can impact the protein in a specific way based on the role of that specific domain. We can also alternatively splice, where we put it together different exons to create slightly different proteins, with slightly different purposes from one gene. So where we kind of finished at the end of the year was we talked about codons and how is the language of RNA turned into a protein, right? And those codons are like words. And we talked about that the genetic code is not your actual DNA, but the genetic code is how do your cells know how to make the protein? What do the words actually mean, and what do they code for? Which amino acids? AUG is always methionine. The other ones that were mentioned there, the UAA, UAG, and UGA are all stop codons. We talked about how the code is universal again, right? With glycolysis, it's evidence of a universal common ancestor. And you have to be able to use this chart, which is basically like you just read, every 3 nucleic acids, starting with AUG usually. This one doesn't, but starting with AUG, every 3 until you hit a stop codon. And we practiced that. And we talked a little bit about the math here associated with, right, 100 amino acids. You have to multiply that by 3 in order to get the total number of nucleotides to code for that 100. So it's about 300. But if you got to stop at 2, you have to add 3 nucleotides to that. So it's 303 if they ask you to stop it as well. Okay, so this is the part where we kind of finished with in the before we left for break. So there are these things called tRNAs, and tRNAs their job is to act almost like sous chefs if the ribosome is the chef. And their job is to bring the specific amino acid to the ribosome. And it uses this part called the anticodon to do that. That'll be the opposite or close to opposite of the codon itself that is in the mRNA. It will be weakly actually base pair, and that's how it knows if it's the proper one. So these are transcribed from special types of genes in the DNA, but they don't code for proteins. They just code for tRNAs. They get reused and reloaded over and over and over again. And there's a thing called the wobble effect, that the third position doesn't matter that much, which is why in the genetic code you tend to see, as we back it up, multiple nucleotide sequences or multiple codons that code for one amino acid, because that third position doesn't really matter all that much. And sometimes there are multiple tRNAs that code for the same amino acid as well. Here's showing the shape of those things. Sometimes we see them as an actual this flattened T-shaped thing. So this is a 3D shaped version of it, and this is just a general model that we use with that anticodon at the bottom. And remember, the anticodon is the opposite of the codon. You do not, it's a common misconception, when you are doing the genetic code, you do not use the anticodon, you always use the mRNA codon. Okay, so don't think you have to do the opposite of it. You just simply read, what does the mRNA say, like AUG, then you look on the chart, you'll say, oh, it's met. So then that'll be met. It's even though that UGA would be on the tRNAs. Okay, so the tRNAs are going to go to the ribosomes and bind the anticodon to the codon, as you can see here down at the bottom. Okay, actually, what's nice is I actually have a pen now that I can use if I can find the one that actually functions. Okay, here we go. So if I turn on the pen, nice. Down here, you can see that happening with the base pairing there. So it always brings in the appropriate amino acid right here. And what's going to eventually happen is they're going to form a bond between these two amino acids, and a bond between the tRNA is going to sever, and all of these are going to move over, and a new one's going to come in, and it just keeps sliding along and kind of doing that. Just showing you that again. Again, what happens is the bond gets severed here, bond is formed there. This one gets kicked out, this one moves over, this one moves into that position, the ribosome slides down, and then another tRNA comes in. And it just keeps doing that until it hits a stop codon, which I think is a pretty simple kind of concept for how it works. So once the tRNA drops its amino acid off, it has to get reloaded with another amino acid. That is called, the enzyme that does that process is called an aminoacyl tRNA synthetase. And as I said, its job is simply to take the proper amino acid, mash it with the tRNA, and form that covalent bond between those two. You can see that kind of happening here. ATP is required for this because it is a endergonic process, so you need free energy to build this, right? You're going from two separate things to one thing. Because it is, again, a non-spontaneous endergonic anabolic process. But you can see how the active site here, how these all fit together. The amino acid fits within the active site, ATP kind of fits in the active site, and so does the tRNA that appropriately matches with that amino acid as well. So ribosomes are where the mRNA and tRNA meet. This is the action site. They're the chef. There's a large and a small subunit that come together, and they're made of both proteins and rRNA. So there's another type of RNA. We've talked about really 4 different types so far. mRNA, or the part that's transcribed, eventually is going to be turned into a protein. Remember, the mature mRNA has been modified in a eukaryote. We talked about tRNAs. We talked about small nuclear RNAs, which are involved in the spliceosome to splice out introns. And now we are talking about rRNAs. And rRNAs are called ribosomal RNAs because their job is to make the actual structure of the ribosome. So if there's a mutation in rRNA, the ribosome is not going to be able to function. You're not going to be able to do the job that the ribosome does, which is to translate proteins, right? So rRNA is ribosomal RNA. If it's messed up, you cannot translate properly, okay? So again, both subunits are made in the nucleus, specifically the nucleolus, which is like a little slightly darker area that's sometimes seen in the nucleus in specific organisms. And they will only join together that large and small when a protein is read. It almost looks like they're like 2 gloves coming together, right? But you have the bottom subunit and the top subunit right here, the small and the large. They come together and kind of fit the mRNA between them. And there are 3 sites in the ribosome, which you don't really have to memorize for the course, though I think it helps you to understand the process. There's the E site, the P site, and the A site. And I will explain to you why it's called that in a second. So eukaryotic and prokaryotic ribosomes are very similar, but they are different. Eukaryotes have larger and more complicated compared to prokaryotes, which are smaller and less complicated. And remember also that mitochondria have their own prokaryotic ribosomes. So they're more like the smaller ones than they are the larger ones, which gives us evidence that they were once free-living organisms, also with the multiple membranes and the fact that they have circular DNA. All right, so as the ribosome moves along that mRNA, as I said, the tRNA will come in and add the matching codon, or it'll match with the codon. It will then bring the appropriate amino acid over, and those bonds will be severed and form, as I mentioned. So the process of building a polypeptide is very similar to the process of transcription. They're, at least, I shouldn't say it's a similar process, but they have the same 3 names: initiation, elongation, and termination. And what is used to build this protein, because this is again an anabolic, endergonic, non-spontaneous process, is we have to get free energy from somewhere. But this time, we actually use GTP instead of ATP. Again, not something that the AP Pop is probably going to ask you about, but if you ever see GTP being used, that's just why. It's just another form of free energy that happens to be used in this process. The other place we used GTP, remember, was in cellular communication when we talked about G proteins. So here's showing you initiation. You see the small subunit attaching to the mRNA. You see the first tRNA come in with methionine attached, because AUG is always the first start codon. You can see that actually right up here, right? If I can I erase with this thing? Probably not. No, won't let me. That's annoying. Okay, I'll just erase like that. Cool. All right. So then what happens? GTP is used. Then you can see the large and the small subunit coming together, and that is initiation. I guess I should have clicked through this. See, this is what happens. It's my first time using this presentation that I've made. I haven't taught with it yet. So that kind of just explained what I just said. Then there's elongation. That's when the codon recognition happens with the tRNA. It matches with its appropriate, its anticodon with the mRNA codon. Then a peptide bond forms where it forms between the adjacent amino acids, right? Between the amino group and the carboxylic acid group with a dehydration synthesis reaction. Think back to Unit 1. Okay, water leaves, we form a peptide bond. Then translocation is everything slides over. The one after the one tRNA is empty, it leaves. Everything slides over, and then it goes. A new one comes in. Now, when we talked about the EPA site before, as I mentioned, why are they called what they are? The E is the exit site. So you can see what's happening in the E site right here is you can see the empty tRNA is leaving. It is exiting. The P site is called that because that is the peptidyl site or the polypeptide site. And you can see that's where the long chain of amino acids is. That's why it's called the P site. And then the A site right here is called that because that's where the new amino acid is coming in, right? So A for amino acid, or amino acyl site, they sometimes call it. And notice as this process happens, we're using GTP, and we can slowly over time assemble that protein, as you can see. So there's 4 here. What happens, you can see that right here, this is going to, this bond gets severed, this one gets formed, this one slides over, right? And then the one that's empty exits. And it just keeps doing that with new amino acids until, as we talked about, what happens? We reach the stop codon. That's termination. So a UAA, UAG, or UGA hits. A release factor protein is going to bind instead of a tRNA, which adds a water at the end instead of amino acid, hydrolyzing it, freeing that polypeptide, and now we have a protein, which will then, as you can see, it all comes in. There's the release factor, which adds water. Everything falls apart. Now we have that free polypeptide that is floating the long chain of amino acids that begins to fold due to the R group interactions that we've mentioned in Unit 1. So if a lot of protein is needed at once, what you can do is attach multiple ribosomes to that mRNA quickly. You saw that in prokaryotes. You can do that in eukaryotes too. It's just you don't have transcription and translation occurring simultaneously. But you can have this happen in eukaryotes as well. So we can make lots of proteins from one mRNA molecule. And that's called a polyribosome because, again, there's multiple ribosomes on one mRNA. So remember, that protein then needs to fold. It comes out straight and uncoiled, but those R group interactions start allowing it to fold, right? Sometimes a chaperone protein, which is a protein that, again, provides a specific, usually hydrophobic microenvironment. So it goes in there, so it folds a little bit, and then it releases it and finishes folding after it gets out of there. So then we have a functional protein. Sometimes proteins, in order to be active, do need post-translational modifications as well. Sometimes they're cleaved or cut, pieces cut off of them to be functional. Sometimes they have to be placed in the proper environment. pH needs to be appropriate. Sometimes they have to bind to another protein to be activated, or a phosphate group has to be added to them in order to be activated. Lots of different things can happen in order to get the protein to do its proper job. So again, just a review from the beginning of the year. Primary structure is the order of the amino acids. That's what the DNA codes for. Goes to RNA, turned to a protein by the ribosome, assembling that primary structure. Secondary structure is how it starts to coil into alpha helices and beta sheets. Remember, not due to R group interactions. Then all of those alpha helices and beta sheets will start to fold and come together due to R group interactions. And then quaternary structure is when multiple subunits or pieces come together to give you the overall functional protein shape. So how do we sense proteins to be made in specific locations? Well, they have these things called signal peptides, and signal peptides are sequences of about 20 amino acids that are found at the end of a polypeptide for a protein that is destined for the endoplasmic reticulum, right, or the endoplasmic membrane for secretion. We talked about that, right? So this is how do we know when a gene reads, when a gene is read, how do we know that that protein is destined to be secreted like a hormone or a digestive protein that has to end up in, or a digestive enzyme that has to end up in your intestinal lumen or something like that, right? These excreted proteins need to be marked with a special, think of it almost like a zip code or an address label. That's what the first 20 amino acids are. If they have that, it's picked up by something called the signal recognition particle. And that's also made of protein and RNA. That grabs a protein being synthesized and it actually brings it to the rough ER. So almost all ribosomes actually start in the cytoplasm. It's only if it starts making the protein and there's a signal peptide present on it, that's when it's grabbed by the signal recognition particle. It brings it to the rough ER. And then it spits that protein out into the rough ER. It gets packaged, shipped to the Golgi, and then finally shipped out of the cell or to the cell membrane, wherever it needs to be. Okay? So we talked about a bunch of RNAs already. Let's review all of the ones we've talked about. So there's messenger RNA or mRNA, right? That carries information specifying amino acid sequences of proteins from DNA to ribosomes, right? There is transfer RNA or tRNA. That's job is to bring the amino acids to the ribosome during translation. There's ribosomal RNA, which actually gives the ribosome its structure. There's the primary transcript, which is the precursor to all of the other ones. Mostly we talk about it, the scope of mRNA, right? That's the stuff that has not been processed yet. We haven't added a 5' cap. We haven't added a poly A tail. We have not spliced the introns out yet. Then there's small nuclear RNAs. Those are involved in the spliceosome, right, for RNA splicing. And then there's signal recognition particle RNA that are involved in recognizing those signal peptides and bringing the protein to the RUFER to be shipped out. So we've seen this picture already, but again, I'm just reviewing this. Translation in bacteria happens at the same time as transcription. Again, notice because there's a polyribosome unit on there, at the same time it's being transcribed, that means it's not being modified before it leaves the nucleus because it's clearly not in the nucleus, as transcription happens in the nucleus in eukaryotes, excuse me. In prokaryotes, transcription happens in the nucleus. In eukaryotes, transcription happens in the nucleus, and in prokaryotes, it happens in the cytoplasm. Whereas translation occurs in the cytoplasm in both. Okay, so remember, if you ever see this, this means it has to be a prokaryote because transcription and translation are occurring simultaneously. And this is the whole process. You can see here RNA is being transcribed from the DNA. Right here, we can see the process of those exons being spliced together and the introns being spliced out. You can see a poly A tail added and a 5' cap was added. It then snakes out of the nucleus through a pore if it's a eukaryote. It goes to the ribosome attaches to the first codon, the AUG, and then it adds methionine, then the large subunit goes, and it just keeps going over and over and over again, using that GTP energy, forming peptide bonds between here and severing this one. Everything moves over until it hits a stop codon, everything falls apart, and then the protein will assemble itself, right, due to R group interactions. Remember, and then those tRNAs can get reloaded using these aminoacyl tRNA synthetases, so they can keep adding amino acids as needed to proteins. All right, that's it for this. If you have any questions, come see me.